Hello friends, this video on Biotechnology Principles Part 20 is brought to you by examfear.com. No more fear from exam. So now once that is done, the next step is going to be the insertion of the recombinant DNA into the host cell, because that is going to be kind of the final step where we want the final recombinant DNA to enter into the host cell. Now as I had mentioned before that this insertion can happen with the help of a lot of techniques like the micropipette method or the gene gun method or electroporation. So all of them help in introducing the recombinant DNA into the host cell. So here the rDNA is transferred into the host cell. Now, as I mentioned before that there can be variety of techniques like the gene gun method, electroporation method and micropipette method. Now, I just discussed them some time back, so I don't think I really need to discuss them again. So, these are the various ways by which this recombinant DNA is inserted into the host cell. Now, transformation of host cells can take place if recombinant DNA is taken up by the cell. Now, here comes the term transformation. So, if you remember, we discussed this, that it is the process where the recombinant DNA, which contains the foreign gene, is inserted into the host cell. Now, whether the transformation will take place or not, that again depends. So, there can be some transformants, there can be some non-transformants. So, if the recombinant DNA is resistant to an antibiotic, so how do we check that, whether the transformation has taken place or not? The same thing, how do we distinguish between the transformants and the non-transformants? So for that purpose, what do? We allow all the entire culture to grow in presence of an antibiotic. For example, we take an antibiotic like ampicillin. So if the recombinant DNA enters into the host cell, in that case the host cell will be resistant to ampicillin. So in that case, now there is one option if recombinant DNA has entered into the host cell. So that is one option. The other option is it has not entered. So if it has entered into the host cell, so in that case what will happen? The host will be resistant to ampicillin, and if it is resistant to ampicillin, then the host cell will grow in ampicillin medium, because the ampicillin antibiotic will not affect it. So if you grow the host cells in an agar plate with ampicillin, then the transformants will be able to grow. So transformants are the one in which the rDNA has entered. So these are the transformants, whereas the ones which could not grow, that is, they are not resistant to ampicillin, that means that the rDNA is not taken up by those cells, and they are the non-transformants. So that this is how you can determine how many of them actually got transformed and how many could not be transformed. So this is how the concept of insertion of rDNA into the host cell take place. So now we will talk about the foreign gene product. So till now we saw that the recombinant DNA was formed. How was it formed? With the help of the vector and the cut piece of foreign DNA, and then they were stuck together with the help of DNA ligase, and that is how we got the recombinant DNA. Then the next step was to insert the recombinant DNA into the host cell. So once that was done using any of the techniques which we described in the previous slide, after that what do we expect? Now our aim is that the foreign DNA should multiply and it should get expressed. Now when I say that it should get expressed, what do we mean when I say that a gene should get expressed? Now let us suppose this rDNA, that is the recombinant DNA, is inserted inside the cell. So this is the host cell and this is the rDNA. So this one is the recombinant DNA and this is the host cell. So inside the host cell it has entered. Now what do we expect? We expect that this gene should get, the foreign gene which is present somewhere here, that should get expressed. When I say that, let us take an example to understand that. When I say that the glow which is present in jellyfish should be also present in a frog, That means I expect the frog also to glow at night, just like the jellyfish. And for that purpose, what do? I extract the gene of the jellyfish and insert it inside the cell of the frog. Now, I want that gene not only to be present inside the cell of the frog, but also to get expressed. That means that glow should be seen in the phenotype of the organism. Now, for these genes to get expressed, appropriate conditions need to be provided. Now, as we saw in our Mendelian genetics, that not all genes get expressed. There are a lot of genes which remain hidden for a few generations. Now, if we want the genes to get expressed, appropriate conditions will have to be provided. When these genes get expressed, then what happens? The target proteins are formed. So basically, the production of target proteins needs to be aimed at a larger scale, because whatever gets expressed, that is because of the synthesis of the corresponding proteins. So we should actually look at synthesizing the right proteins at a large scale, and that is where we talk about a device called bioreactors. So what is our aim in this case? Synthesis of the desired proteins on a large scale. And how do we do that? And there comes the concept of bioreactors. So now we will spend some time to see what are bioreactors. So bioreactors are devices in which large volumes of culture can be processed to form desired products. Now, what are the desired products in this? So, these devices, they are not exactly devices, they are like vessels. You can just imagine them as large-sized vessels. Now, when we say that in this device, desired products can be formed. So, what are those desired products? Desired products like enzymes, which are useful to us. There are several medicines which are produced. So all those are desired products. So we want to produce them, and for that we need to do a large volume of culture. And culture of what? Of organisms. Some organisms are cultivated in these devices. Organisms like plants or animals or microbial cells. So they are cultivated on a large scale because large space is being provided. So a lot of them can be cultivated so that a good amount of desired products can be formed. So here appropriate environment is provided for optimal growth and metabolic activity of the organisms, because it is due to the metabolic activity of these organisms that all these desired products can be formed, because we are interested in their gene, and to get that gene, that organism should be healthy enough, it should grow, it should undergo its metabolic activities. So all those things should happen. So going to the types of bioreactors, well, there are different types of bioreactors which are present. For example, stirred bioreactor, photobioreactor, membrane bioreactor, airlift bioreactor, mist bioreactor. So they are all different. I mean, there are many other different types of bioreactors as well, and all of them, their main purpose is the same thing, that is they will provide an environment where the culture can be grown so that desired products can be formed. But all of them will have some different structure, they will make use of some different form of energy, so that is how they are all different. Well, here we are not going to talk about each of these type of bioreactor in detail, but still we will just touch upon the stirred bioreactor. So here you can see the picture of a stirred bioreactor. So we will talk about stirred bioreactor for some time. So here you can see these are the different parts of the reactor. So this is the motor which will help in mixing, that is this needs to be rotated, as you can see the arrows. So it actually rotates, and that rotation happens with the help of the motor. So here you have the culture broth. So this is where the organisms are cultivated. You have an opening here from where the sterile air gets in. Sterile means pure, which is free from any sort of contamination. So here again you have another opening where you provide steam for sterilization. Here again you have another controller, and this controller is to control the pH. So pH is being controlled from here. So when I say pH, increasing or decreasing the pH can change the medium from acidic to basic. So that's how the medium is going to be, that is controlled by this pH control. You also have a temperature controller to control the temperature inside. So there are lots of controls. If you see, pH control is there, temperature control is there. In fact, you have a foam control system as well. So here you can see these blades which you see here, these are nothing but the foam breakers. So these are the foam breakers. So that ways you have so many controls here in this. So if you see, it is cylindrical in shape and it has a curved base. So this shape and this structure enables the mixing of the reactor contents really well. It also facilitates the availability of oxygen. That is where you see here you have an opening. It also has a lot of control system, as I said, the pH control system, the foam control system, temperature control system, agitator system. That is this, you can see the blades and all, when it moves, it agitates the entire content of the reactor. Oxygen supply system. So all these systems are there in place. So what happens as a result of this entire thing? The result is a larger scale production of the recombinant proteins, because that is what we are aiming at. We want the right kind of proteins to be formed in large amount, and for that purpose we make use of these bioreactors. Now we will not get into the detail of the functioning of different parts of the bioreactor. This was just to give you an idea that how these kind of instruments can be useful in production of proteins. And then the final step, that is downstream processing. So now once your proteins are being synthesized, you are almost there. So now it is like the last step where you give the finishing touch to your product before you start marketing it. So that is when we talk about downstream processing. So this is the process which ensures purification of the biosynthetic products. So your products are already being created, like your enzymes are already being created. So whatever has to be created is already created. You just need to ensure its final purification round. So in this round, what you have to do is you need to ensure that all the unwanted cells or debris are all removed, either by filtration or sedimentation or centrifugation. So all these methods can help in removing the unwanted materials which might be present in the final product. So removal of unsolubles, so their filtration or sedimentation can help. Filtration or sedimentation. So I hope by now you know what is filtration, what is sedimentation. So in sedimentation, what happens? The heavier particles will settle down, so the insolubles will settle down, and the good product will be there on the upper side. So that way you can separate it. Product isolation, so the removal of solvents like water, etc. should be removed, and only the product should be taken out. So the product needs to be isolated from all other solvents. Product purification. Now we need to separate the particles which might very closely resemble the final product. Let us suppose the final product are some enzymes. Now there are some other particles which are unwanted, but they resemble enzymes in their structure a lot. So we also need to ensure that those kind of particles are also removed. Product polishing. Packaging the product is also very, very important, so that it is easily transportable, it is quite stable. The packaging should be proper so that it doesn't break or it doesn't fall or it doesn't get contaminated. So it has to have a stable packaging which is transportable. The product need to be sterilized. It should not have any sort of contamination. So all these final touches need to be given to the final product, and then there you are. So you get the final product. So now here you saw the entire process of genetic engineering step by step. So you started from identifying the right DNA or a desired gene in the DNA. So from there, gradually you used that gene, you transferred it to some other organism, and then you finally reached out having a new product altogether. So this is how genetic engineering helps. I mean, this is how the entire process of genetic engineering works. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.